What you're seeing now are Calyobacter crescentis cells under a microscope. These cells are termed Prosthecate bacteria, also known as Stalked bacteria. The term describes a phenotypic group of organisms that are characterized by their appendages named Prosthecate. Prosthecate, also known as stalks, are neither pili nor flagella. They are instead extensions of the cell wall and membrane, typically containing a narrow core of cytoplasm. Stalks are for nutrients uptake. They increase the surface area to volume ratio of the bacteria, helping them scavenge nutrients in their aquatic nutrients-poor habitats. The appendages also increase the buoyancy of the cells, keeping them close to the water-air interface of, say, lakes and swamps. This is crucial to aerobic respiration, which many Prosthecate bacteria species undergo. Prosthecate bacteria vary greatly in their taxonomy. Although Prosthecate bacteria are predominantly alpha-proteobacteria, some stalked bacteria are not. For example, Gallionella ferruginea is beta-proteobacteria, and Nevskia is gamma-proteobacteria. Prosthecate bacteria of the other classes have interesting differences in their stalks. Prosthecate alpha-proteobacteria tend to have thin and long stalks, while stalked bacteria of other classes have slime-producing and twisted stalks. Some Prosthecate species exhibit a dimorphic cell cycle, which means that the daughter cells have different morphologies. Gram-negative Calyobacter crescentis has a distinctive morphology for each stage of its cell cycle. Cell division for this species results in a stalked daughter cell and a swarmer daughter cell. A stalk is required for C. crescentis reproduction. If a swarmer daughter cell does not have a stalk, C. crescentis produces only one daughter cell capable of reproduction per division. The purpose of the swarmer cell is to find livable niches in their hostile environment, and after finding such places, differentiates into stalked cells and starts reproducing. C. crescentis is a widely studied bacterial species, and its easily observable life cycle allows detailed understanding of bacterial differentiation, bacterial development, and cell cycle regulation. While it is known for its unusual cell cycle, it has other fascinating near-unique features. Calyobacter forms biofilms which stalked cells are attached to permanently, while swarmer cells can leave. C. crescentis cells kill themselves through an endonuclease-based apoptosis-like mechanism if they face DNA damage beyond repair. DNA of the dead cells is released and promotes dispersal of swarmer cells, increasing the newborn's chance of survival. Similar to C. crescentis, Ascacalus beneficetus is a gram-negative aerobic organism that also undergoes asymmetric cell reproduction. However, A. beneficetus differs from C. crescentis in its stalk. For example, while C. crescentis has the holdfast adhesive as the tip of its stalk, which lets itself stick to surfaces, the A. beneficetus stalk does not have holdfast. Like many other Prosthecate bacteria, A. beneficetus is an oligotroph, meaning that it tolerates nutrient scarcity, able to survive in places where organic materials are available in low quantities. Known as iron bacteria, Gallionella can be found in many different environments, such as freshwater, mineral springs, and shallow brackish waters, which are saltier than freshwater but less salty than seawater. They use inorganic chemicals for energy generation. Waterworks, wells, and drains are places with the best prospects of finding the bacteria in abundance. Perhaps the presence of the bacteria is best shown by deposits of iron oxide compounds, which clog the pipes and drains. Gallionella ferruginea is a good example, as it is found in the environments listed before. G. ferruginea generally is found in abundance in pure iron-bearing waters with low traces of organic material. It is a chemolithotrophic species that oxidizes iron. It thrives in environments in where there is sufficient carbon, oxygen, phosphorus, and nitrogen. Electrochemical and molecular properties of the bacteria allow it to remove dissolved iron and water. This produces an insoluble precipitate of ferric hydroxide and a bacteriologic iron oxide precipitate.